My name is Nikolai Slavov. I'm a co-founder at Parallel Square Technology Institute.
I'm Harrison Specht. I did my PhD in the lab of Nikolai Slavov. We worked on single-cell proteomics and scaling technologies. That's kind of what led us to the conception for the FRO, Parallel Square Technology Institute.
I have the privilege of working on one of the most impactful problems in science, which is understanding how proteins function in health and how they dysfunction in disease.
If genes are the sort of the blueprint in biology, proteins are the effectors, the actors that make biology happen.
The proteome remains one of the biggest frontiers in science that is so challenging to study and overcome that we still haven't detected all of the proteins in the human body, and we are still very far from understanding how all of these proteins cooperate together. And one of the important milestones along this journey was the development of mass spectrometry as an approach. It is a way to measure the masses of molecules in their ionized form very, very accurately and precisely. And then we can break those molecules and measure the masses of all of the pieces. And we end up with something like the pieces of a jigsaw puzzle. And if we have all of the pieces, we can put them together and understand what the molecule was.
These mass spectrometers, incredibly powerful, but also incredibly expensive. But if we can increase the amount of samples that can be processed in that hour, that means each sample costs a fraction of that.
We need to acquire more data points. It's as simple as that. And we figured out how we can simultaneously analyze peptides derived from multiple samples.
We are developing the tool that will allow us to find as many proteins as possible, but more on an emphasis of as quickly as possible. We're hoping to do that same order of magnitude, several order of magnitude decrease in the cost of processing a sample for proteomics that has happened for genomics in a fraction of the time.
Success means that everyone around the world who studies biomedical research biology is able to directly analyze proteins in ways that are very scalable, that don't compromise depth of coverage, quantitative accuracy, and that this analysis fundamentally changes the type of inferences and the confidence that people can make from the data. Understanding this has the potential to completely transform our ability to develop effective therapies and what makes us alive, what makes us human.